Let's get into some science. I'm Brian Ross. I'm the head of biochemistry at the Quantum Biology Institute. We're interested in exploring quantum effects in biology. So one thing we're really interested in is how magnetic fields affect proteins. And to study this, we are developing new instruments like the Bacterioscope. The Bacterioscope is a bacterial plate imager that allows us to manipulate magnetic fields and study the effects of proteins that are expressed in the bacteria. The ultimate goal of the Bacterioscope is to detect phenomena that can only be explained through quantum mechanisms that we can further study using our quantum microscope that we are developing. So we've already run some experiments on the Bacterioscope, and I'm very excited to show you how we analyze that data and what some of our preliminary results are showing us. In this experiment, I take a gene expressing a protein that is magnetosensitive, and I insert the DNA into the bacteria and have the bacteria express that protein. We then take the bacterial plate and image it with our bacterioscope. And as we image it, we modulate the magnetic field. And this particular protein is fluorescent, and its fluorescence changes in the presence or absence of external magnetic fields. And our bacterioscope allows us to measure those changes, what we call the magnetic field effect. So let's get into the data analysis. And if this feels a little technical, no worries, feel free to leave a comment, and we're happy to answer any questions you may have. So let's look at some data that we collected from the Bacterioscope. What we see here is an image of bacterial colonies that are on an agar plate. Each of these colonies are expressing a magnetosensitive protein, and that protein is fluorescent. And so what we're capturing here is the fluorescence from each of the colonies. We ran a script to segment the image into regions of interest that capture each of the colonies, and then we measured the average fluorescence over time in each of those images. When we collected these images, we pulsed magnetic field. And so what you can see if we skip through the data is that the fluorescence increases and decreases as we manipulate the magnetic field. With these different regions of interest, each representing a colony, we could quantify the fluorescence over time as we change the magnetic field. Then we analyze that data to extract just the magnetic field effect, that is the change in fluorescence that's only due to the magnetic field. And how we did that is first, I plotted here for every colony the fluorescence trace, and we see that in black here. The teal represents magnetic field on and the white magnetic field off. And you can see there's a decrease in fluorescence in the presence of the magnetic field that reverses when we turn off the magnetic field. Did that for all of the colonies. And then we fit a curve of just the points when the magnetic field was off to represent what the fluorescence would have looked like if we didn't have an external magnetic field. And that way we can isolate just the magnetic field effect. So we did that by calculating what's called a residual. And so we see the residuals here. So that removes any effect of photo bleaching or photo damage from the light or other effects that could change the fluorescence in the absence of an external magnetic field. In this particular experiment, we ramped the external magnetic field that we applied from 100 millitesla down to 5 millitesla. So this is 100 millitesla, 95, 90, etc. And we see a decreased size in the magnetic field effect with respect to the strength of the magnetic field. So these are plots of the averages across the plate. And so this is the fluorescence trace in black. And this red curve represents what the fluorescence would be in the absence of the external magnetic field, which we found using exponential fit. And then we calculated residuals, which represent the magnetic field effect on its own. We then quantified the size of the magnetic field effect for each of the magnetic field strengths. And we plotted them, the magnetic field effect, in terms of the percentage of the fluorescence, plotted against the magnetic field strength. We repeated this experiment for other ranges of magnetic fields. In this case, we're starting at 10 millitesla, going all the way down to 0.5 millitesla. And we see something very, very interesting. At the higher magnetic field strengths, we see a decrease in fluorescence in the presence of the magnetic field. But when we get to very, very low magnetic fields, we actually see an opposite. We see a very slight increase. So if we plot the entire scale from 0.5 millitesla all the way up to 100 millitesla, this is what the curve looks like. If we zoom in at the initial part, what we see here is that at very low magnetic fields, the effect on fluorescence is positive. So if we add the external magnetic field on top of the magnetic field of the Earth, we actually see an increase in fluorescence. But somewhere around 2.5 to 3 millitesla, that effect changes to be negative. And we see that it becomes increasingly negative as we increase the magnetic field. And we see that that trend continues. But at some point, the magnetic field effect starts to plateau. And at very high magnetic fields, we see that diminishing returns, where an increase in magnetic field strength doesn't yield as large of a change in magnetic field effect. There are no classical models to explain this data, but it's very consistent with the radical pair mechanism, our quantum model for explaining magnetic field effects in proteins.